It is 1969. China is on fire, not with war, not with famine, but with ideology. Universities have been shut down. Laboratories sit empty. Scientists have been sent to work in rice fields to confess their bourgeois sins before crowds of Red Guards. And Mao Zedong's Cultural Revolution, the pursuit of Western-style science, is not just discouraged, it is dangerous. And yet, somewhere in Beijing, a woman is saying goodbye to her children. She doesn't tell them where she's going. She can't. She has signed a secrecy agreement. She doesn't know when she'll be back. It will turn out to be years. Her youngest child is barely a toddler. Her eldest is 4. She hands them to relatives and walks out the door.
Take care.
Her name is Tu Youyou. She is 39 years old. She holds no doctorate. She has never studied abroad. By the standards of international science in 1969, she is almost invisible. But someone has chosen her for something extraordinary. The project has no public name. It is known only by a date, the 23rd of May, Project 523. 500 scientists scattered across China, working in total secrecy, under military oversight, during a time when science itself has been declared an enemy of the revolution. What are they looking for? A cure for a disease that, at that very moment, is killing more soldiers in the jungles of Vietnam than bullets ever could. And the answer, though none of them know it yet, is not in a laboratory. It is in a book, written 1,600 years ago by a physician who had never heard of a parasite, never seen a microscope, and never imagined that one day his words would save hundreds of millions of lives. This is not a story about a Nobel Prize. This is a story about what happens when modern science runs out of answers and history steps in.
Let's start with a number, 608,000. That is how many people died from malaria in a single year, 2022, according to the World Health Organization. Not in ancient history, not before modern medicine existed. Last year, the year before. Every year, like clockwork, across 85 countries, malaria kills. Mostly children, mostly in sub-Saharan Africa, mostly in places where the disease has been allowed to run unchecked for generations. Now, here's another number, 340. That is the year, AD 340, when a Chinese physician named Ge Heng wrote down a recipe for treating malarial fevers. A handful of a plant called Qinghao, soaked in cold water, the juice wrung out and swallowed whole. Between those two numbers, 340 and 2022, lies one of the strangest, most consequential stories in the history of medicine. A story about a disease that has plagued humanity for at least 5,000 years. About a secret military operation launched in the middle of a cultural revolution. About a woman who read a 1,600-year-old recipe and understood something that hundreds of modern chemists had missed. About a drug that the World Health Organization refused to endorse for 2 decades and then, quietly, made the centerpiece of global malaria treatment. By the end of this video, you will understand not just what Artemisinin is and how it works, you will understand why the world took so long to accept it, and you will understand what that delay cost. Because the cure was already written, it just needed someone willing to read the old books.
To understand why Project 523 existed, you have to understand what was happening in the jungles of Southeast Asia in the 1960s. The Vietnam War is often remembered through the lens of politics, the protests, the draft, the fall of Saigon. But on the ground, in the dense rainforests where American and North Vietnamese soldiers were actually fighting, there was a second war happening, a quieter war, and it had no ideology, no allegiances, and no mercy. Malaria.
Plasmodium falciparum, the most lethal of the malaria-causing parasites, thrives in exactly the kind of warm, wet, mosquito-dense environment that both sides were now living in. Soldiers who survived ambushes, firefights, and artillery strikes were being felled by a microscopic parasite injected by a mosquito bite they never even felt. By the late 1960s, estimates suggested that malaria was incapacitating more troops on both sides than combat wounds. Some units were losing half their fighting strength not to enemy fire, but to fever, chills, and organ failure. And there was a new problem, a terrifying one. Chloroquine, the drug that had been the standard malaria treatment since World War II, was no longer working. Plasmodium falciparum had mutated. The parasite had evolved resistance, first appearing in Southeast Asia in the late 1950s, spreading through the region through the 1960s. Soldiers were taking their chloroquine tablets faithfully, and the malaria was breaking through anyway. The drug that the world had relied on for 20 years had become unreliable, and in some regions, useless. The United States military recognized this as a crisis. They invested heavily in finding a solution. The Walter Reed Army Institute of Research launched one of the largest drug screening programs in history, eventually testing over 200,000 chemical compounds in search of a new anti-malarial. It was a triumph of industrial-scale modern science, vast resources, cutting-edge technology, thousands of researchers. They found some useful compounds, but nothing that would solve the problem cleanly, nothing that would change the equation. Meanwhile, the North Vietnamese were facing the same crisis, with none of America's resources. Ho Chi Minh turned to his most powerful ally. In 1964, Ho Chi Minh sent a formal request to Mao Zedong. China needed to help solve the malaria problem killing North Vietnamese soldiers. Mao agreed. Zhou Enlai, China's premier, coordinated the response. Minute meetings from that period described the goal plainly: to keep the Allied troops combat ready. On the 23rd of May, 1967, in the middle of the Cultural Revolution, representatives of China's National Science Commission and the military gathered at a hotel in Beijing. The meeting was secret. The project they launched that day would carry no public name, only its launch date, Project 523. More than 500 scientists were eventually recruited. They were divided into research streams, some pursuing new synthetic chemical compounds, others investigating traditional Chinese medicine, others working on insecticides and prevention, all of them working under strict military secrecy. None of them able to publish, none of them able to speak about their work publicly. The paradox at the heart of this operation was almost absurd. China was simultaneously telling its scientists that Western-style science was ideologically suspect and secretly ordering those same scientists to conduct cutting-edge biomedical research. In the Cultural Revolution, universities were shut, intellectuals were persecuted, and scientific journals had largely stopped publishing. And yet, 500 researchers were being asked to solve a problem that had defeated the entire Western pharmaceutical establishment. The only resource they had that the Americans didn't? 2,000 years of written medical tradition. And in January of 1969, they asked a 39-year-old researcher named Tu Youyou to lead the part of the project that would dig into it. The decision that would change everything had just been made, though no one in that room could possibly have known it yet. Tu Youyou was not a famous scientist. She had graduated from Beijing Medical University's School of Pharmacy in 1955 and had spent the years since then at the Institute of Chinese Materia Medica, studying both traditional herbal medicine and modern chemistry, developing what would turn out to be a uniquely valuable combination of skills. She could read classical Chinese medical texts. She understood organic chemistry. She was fluent in both worlds at a time when those worlds were treated as opposites. From 1959 to 1961, she had undertaken a special training program in classical Chinese medicine, reading ancient texts, shadowing practitioners, learning theories that had been accumulating for 2 millennia. She later described this as one of the most formative experiences of her career. When the Project 523 directors came to her institute in January 1969, they weren't looking for a celebrity. They were looking for someone who could do something specific, search the entire recorded tradition of Chinese herbal medicine for any historical treatment of malarial fevers, and then test those treatments using modern scientific methods. Tu Youyou accepted. She said goodbye to her children and went to work. She began the way any serious scholar would, by reading everything. Over the following months, she and her small team combed through ancient medical texts, folk remedy collections, and historical manuscripts. They consulted practitioners of traditional medicine across the country. They gathered information from every possible source, written and oral, official and informal. In the end, Tu compiled a report titled Anti-Malarial Collections of Recipes and Prescriptions. It contained more than 2,000 possible treatments distilled from the historical record and summarized 640 specific prescriptions that seemed relevant. Among those 640 was a plant called qinghao, the Chinese name for Artemisia annua, or sweet wormwood. A slender, feathery plant with small yellow flowers, unremarkable to look at, with a sharp, slightly bitter smell. It had appeared in Chinese medical texts for centuries as a treatment for various fevers. But when the team began testing, qinghao initially did not stand out. Their extracts showed inconsistent results, sometimes promising, often not. Other research groups within Project 523 had also looked at qinghao and moved on. It seemed like a dead end. Tu Youyou kept returning to it. Something about the historical record nagged at her. The plant kept appearing. Different dynasties, different physicians, different texts. Qinghao and fever kept showing up together. There was a pattern there that the laboratory results weren't capturing. And then she found a single line in an ancient text that changed everything.
The text was called the Zhouhou Beiji Fang, roughly translated as the Handbook of Prescriptions for Emergency Treatments. It was written by a physician and scholar named Ge Hong during the Eastern Jin dynasty, around the year 340 A.D. Ge Hong was not a fringe figure. He was one of the most respected medical minds of his era, a Taoist scholar and alchemist, a physician who synthesized the medical knowledge of his time into practical handbooks intended for ordinary people who might not have access to a doctor. His Zhouhou Beiji Fang was essentially an ancient first aid manual, compact, practical, designed to be used in emergencies. And in it he had written about qinghao. His instruction was precise. Take a handful of qinghao, he wrote, immerse it in 2 liters of water, wring out the juice, consume it in its entirety. No boiling, no heating, cold water only. Wring and drink. Tu Youyou read that line and stopped, because the standard extraction method that her team and every other research group had been using involved heat. They were boiling or heating the plant material to extract its active compounds, the way modern pharmaceutical extraction typically works. High temperatures drive out more material. It's more efficient. But Ge Hong had specifically said cold water. Why would a physician in the 4th century insist on cold water? He had no concept of chemical stability, no understanding of molecular structure. He was describing what worked, empirically, through centuries of observation and practice. He didn't know why cold water mattered, he just knew that it did. Tu Youyou suspected that whatever was active in qinghao might be heat-sensitive. that their extraction process might be destroying the very compound they were trying to isolate. She redesigned the experiment. Instead of ethanol extraction at boiling temperatures, she switched to ether, a solvent with a boiling point of just 35 degrees Celsius, much lower heat, much gentler process, closer in its own way to what Ge Hong's cold water method was achieving 1,600 years earlier. The result was unambiguous. Tu Youyou's laboratory record lists Artemisia annua L as experiment number 191. Parasite inhibition ratio 100%. Not 90, not partial, 100. On the 8th of March 1972, Tu Youyou stood before a major national meeting of Project 523 participants from research institutes across China and presented her findings. The title of her presentation carried the required political framing of the era. She called it Discovery of Anti-Malarial Chinese Herbal Medicine under the Guidance of Mao Zedong's Thoughts. The ideology was the wrapper. Inside the wrapper was a scientific result that none of the other 500 researchers in the program had achieved. The room understood immediately what it meant. Within months, teams across China shifted focus. King Hao became the priority. Tu's team moved to isolate and purify the active compound, a process that required further refinement to remove toxic impurities from the extract. By late 1972, they had produced pure artemisinin crystals. Chemical structure was determined by 1975 and published in 1977, anonymously, as the egalitarian ethos of the Cultural Revolution required no individual names on scientific papers. In 1972, Tu led a clinical trial on Hainan Island, a malaria endemic region in southern China. She had first taken the extract herself to evaluate its safety. As head of the research group, she said later, she felt it was her responsibility. 21 patients were treated. Efficacy was between 95 and 100%. The answer to a 1,600-year-old question had been confirmed in a mosquito-infested island clinic. But almost no one outside China would know about it for years, because Project 523 was secret. And what happens when a secret operation makes a world-changing discovery? The world doesn't find out. Not yet.
Before we follow Artemisinin out into the world, it's worth pausing to understand what makes it so extraordinary as a molecule. Because the mechanism by which it kills malaria parasites is unlike almost anything else in pharmacology. To understand it, you first need to understand what malaria actually does inside the human body. When an infected Anopheles mosquito bites you, it injects Plasmodium parasites into your bloodstream. These parasites make their way to your liver, mature, and then re-enter the bloodstream in a form called merozoites. The merozoites do something elegant and brutal. They invade your red blood cells. Once inside a red blood cell, the parasite makes itself at home. It begins consuming hemoglobin, the protein that carries oxygen through your blood. As it digests hemoglobin, it releases a toxic byproduct called heme, which contains iron. For the parasite, managing this iron is a critical challenge. It has evolved mechanisms to sequester and neutralize the heme before it damages the parasite itself. It is, in essence, mining your blood for nutrients while carefully disposing of the toxic waste that process generates. This is where Artemisinin enters, and it exploits exactly that process. Artemisinin belongs to a chemical class called sesquiterpene lactones. What makes it pharmacologically unique is a structure within the molecule called an endoperoxide bridge. Two oxygen atoms bonded to each other in a configuration that is exceptionally reactive under the right conditions. And the right conditions exist precisely inside a malaria-infected red blood cell. The free iron released during the parasite's digestion of hemoglobin reacts with artemisin's endoperoxide bridge. This reaction generates what chemists call free radicals, highly reactive molecular fragments that cause rapid, cascading damage to the parasite's cellular machinery. Proteins are disrupted, membranes are damaged. The parasite's own metabolic process, the very process it was using to feed and grow, becomes the trigger for its destruction. In other words, artemisin turns the parasite's iron against it. The parasite is killed by a weapon it inadvertently loaded itself. This mechanism is also why artemisin acts so rapidly, because it exploits an active, ongoing metabolic process inside the parasite. It begins working almost immediately after entering the bloodstream. Fever breaks quickly. Parasite counts drop fast. Patients often feel dramatically better within 24 to 48 hours, faster than with any other anti-malarial treatment known at the time. There is one more detail worth understanding: the specificity of this reaction. Normal, healthy red blood cells contain very little free iron in the form that triggers artemisin's endoperoxide chemistry. The reaction happens predominantly inside infected cells, cells where the parasite is actively digesting hemoglobin and releasing iron. This is one of the reasons artemisin has a relatively favorable safety profile. It is targeting the parasite's own chemistry in the parasite's own environment. Ge Hong had no concept of any of this. He could not have known about parasites, iron chemistry, free radicals, or endoperoxide bridges. He knew that Qing Hao, prepared cold, reduced fever in patients who likely had malaria. He observed it, he recorded it, he insisted on the cold preparation, preserving, without knowing it, the molecular structure that made the whole thing work. The low-temperature insistence was not mystical, it was empirical. 16 centuries of empirical observation compressed into a single practical instruction. When Tu Youyou switched from hot ethanol to cool ether extraction, she was, unknowingly at first, honoring that instruction. She was getting closer to what Ge Hong's cold water was already doing, preserving the endoperoxide bridge that everything depended on. It is, in retrospect, one of the most elegant examples in the history of medicine of traditional observation and modern mechanism converging at exactly the right point. The bridge between a 4th-century Chinese physician and a 21st-century anti-malarial drug is not mysticism, it is chemistry and the careful preservation of a single molecular bond across 1,600 years.
The artemisinin crystals existed. The clinical data existed. The mechanism, though not yet fully understood, was real and reproducible, and almost no one outside China knew. Project 523 operated under strict secrecy rules throughout its existence. Researchers could share findings within the working groups but could not publish publicly. The Cultural Revolution's political constraints made individual attribution on scientific papers unacceptable anyway. Tu Youyou's landmark 1977 publication appeared without her name. It was authored simply by the Qinghaosu Anti-Malaria Coordinating Research Group. No individual scientist, no institution, no discovery story, just results stripped of their human context by ideology. The first publication in English, the language that would allow the global scientific community to engage with the findings, appeared in December 1979 in the Chinese Medical Journal. It attracted some attention. It brought collaboration inquiries, but the research remained largely closed to non-Chinese scientists. The world was looking, but China wasn't quite ready to open the door. The breakthrough in international visibility came in 1981. A researcher named Keith Arnold, based at the Roche Far East Research Foundation in Hong Kong, had been working on mefloquine, another antimalarial compound developed by the American military's research program. Arnold had heard about the Chinese artemisinin work and arranged a collaboration with Chinese researchers, including Guoqiao Li. In 1982, they published a comparative study in The Lancet, one of the world's most prestigious medical journals, showing that artemisinin outperformed mefloquine against chloroquine-resistant Plasmodium falciparum. By Arnold's own account, the Chinese drug beat his. The paper attracted attention. The World Health Organization began to engage. Slowly, painstakingly, the international research community began to study artemisinin and confirm what Chinese scientists had known for a decade. But formal WHO endorsement moved with the speed of an institution designed for caution. The first WHO treatment guidelines formally recommending artemisinin combination therapy, ACT, as the first-line treatment for malaria did not appear until 2001. Nearly 30 years after Tu Youyou stood in a room on Hainan Island and watched 21 patients recover. The delay had consequences. In the 1990s, as chloroquine resistance spread through sub-Saharan Africa and malaria mortality rose sharply, particularly among children, artemisinin was available and proven, but not yet universally endorsed or accessible. The cost of institutional caution in this case was measured in human lives. Eventually, the world caught up. Today, artemisinin combination therapies are the standard first-line treatment for malaria worldwide, recommended by the WHO and distributed across the globe. The drug is estimated to prevent hundreds of thousands of deaths every year. The Lasker Foundation in 2011 called the discovery of artemisinin arguably the most important pharmaceutical intervention of the last half-century. In 2015, the Nobel Committee in Stockholm awarded the Nobel Prize in Physiology or Medicine to Tu Youyou, making her the first mainland Chinese scientist to receive a Nobel Prize in a scientific category and the first female citizen of the People's Republic of China to win a Nobel in any field. She accepted it with characteristic restraint. She called artemisinin a gift from traditional Chinese medicine to the world. She deflected credit to her colleagues, those living and those long dead. Project 523 was officially terminated in 1981. It had lasted 13 years, recruited more than 500 scientists, and produced, in addition to artemisinin, several other antimalarial compounds still in use today, including lumefantrine and pyronaridine. But the story does not end in Stockholm in 2015. Because on the other side of the world, in the river deltas and highland villages of Cambodia, Thailand, and Myanmar, something troubling had begun to appear in the data. Plasmodium falciparum, the same parasite that had learned to resist chloroquine in the 1960s, was showing signs of a delayed response to artemisinin. Not full resistance, not yet, but clearance times were increasing. Parasites that should have been gone in 48 hours were lingering. Mutations in a gene called Kelch13 were being identified as markers of reduced sensitivity. History, it seemed, was not finished repeating itself. The same pattern that had created the crisis of the 1960s, a reliable drug, growing resistance, the gradual erosion of a treatment's effectiveness, was beginning to trace itself again. And this time, there is no ancient manuscript waiting with an answer. No 1,600-year-old recipe to unlock. This time, the world will have to find the next answer on its own.
Before we close, there are some important things to say clearly, because this story, as remarkable as it is, can be misread in ways that cause real harm. Artemisinin works. That is not in question. Decades of rigorous clinical trials and hundreds of millions of treated patients confirm it. But how it works and how it must be used matters enormously. First, artemisinin is never used alone. This is not a minor technical point. When artemisinin is used as a monotherapy, by itself, without a companion drug, it clears parasites rapidly but incompletely. Patients feel better quickly, sometimes within a day or two, and may stop treatment before the parasite is fully eliminated.
Surviving parasites exposed to artemisinin but not killed are exactly the population most likely to develop resistance. Artemisinin combination therapy, ACT, pairs artemisinin with a longer-acting partner drug specifically to prevent this. The artemisinin hits fast and hard. The partner drug cleans up what remains. Used together, they are far more effective and far less likely to generate resistance than either would be alone. Second, the raw plant is not the drug. Artemisia annua grows in many parts of the world. Teas and infusions made from the plant have been promoted in some circles as treatments for malaria. And during the COVID-19 pandemic, similar claims were made about other conditions. The concentration of artemisinin in raw plant material is highly variable and generally low. The dose delivered by a tea or infusion is unpredictable, almost certainly subtherapeutic, and entirely uncontrolled. A person treating active malaria with Artemisia tea instead of ACT is not getting treatment. They may be getting a fraction of the required dose, enough to feel marginally better, not enough to clear the infection, and potentially enough to contribute to resistance. Third, the emergence of resistance is a genuine, documented threat. As mentioned, artemisinin-tolerant strains of Plasmodium falciparum have been identified in Southeast Asia, and more recently, concerning signals have appeared in parts of Africa. This is not a theoretical worry. Researchers are actively working on next-generation antimalarials, alternative combination regimens, and yes, re-examining both synthetic and natural compound libraries for the next breakthrough. The urgency is real. And fourth, perhaps most importantly for how we understand this story, the fact that artemisinin came from traditional Chinese medicine does not mean that traditional medicine is equivalent to evidence-based medicine, or that ancient recipes should be taken on faith. What this story demonstrates is something more specific and more interesting, that empirical observation, accumulated over centuries, can preserve genuinely useful information in forms that modern science can validate and refine. Ge Hong didn't understand why cold water Qinghao worked. He recorded that it did. That distinction matters. The value was in the observation and in the willingness of a modern scientist to take it seriously enough to investigate it rigorously. Tu Youyou didn't skip the science. She used the ancient text to point the science in a direction it had missed. That is the lesson. Not that old books have magic answers, but that old books sometimes hold data, encoded in recipes, in practical instructions, in the accumulated observations of generations of healers that modern methods can unlock.
In the year 340 AD, a Chinese physician named Ge Hong sat down and wrote a handbook. He was trying to help ordinary people survive in a world without hospitals, without pharmacies, without any of the infrastructure we take for granted. He wrote down what worked, practically, simply, without embellishment. Take a handful of Qinghao, soak it in cold water, wring out the juice, drink it. He didn't know about parasites, he didn't know about iron chemistry, or free radicals, or endoperoxide bridges. He just knew, because he had seen it or learned it from those who had, that this plant, prepared this way, could reduce the fever that we now call malaria. 1,600 years passed. Empires rose and fell. China changed beyond recognition. Dynasties, revolutions, wars, a cultural revolution that tried to erase the very idea of inherited knowledge. And somewhere in the chaos, a secret military project commissioned by Mao Zedong sent a 39-year-old pharmacologist into a library to read the old books. She read them carefully. She found one line. She changed her extraction method, and in a clinical trial on a malaria-stricken island, 21 patients recovered. The world took another 3 decades to fully accept what she had found. By now,
Artemisinin combination therapies have treated hundreds of millions of people. The WHO estimates that artemisinin-based treatment has saved millions of lives, with some estimates suggesting the drug has prevented more than 100 million deaths since it entered widespread global use. Every year, it prevents roughly 600,000 more. 600,000 people who are alive because of one line in a 4th century emergency handbook. There is something humbling in that number, and something worth sitting with. We tend to think of medical progress as a straight line forward, each generation building on the last, moving away from the primitive and toward the sophisticated. And in many ways that's true. But artemisinin reminds us that the line is not always straight, that sometimes the most sophisticated thing a scientist can do is read carefully, take seriously what has been observed across centuries, and ask, what if this is pointing at something real? The answer was always there. It just needed someone willing and rigorous enough to find it.